Thank you for having me today. I'm Dr. Emma Leishman, the Technical Manager of the Advanced Analytics team at Avomeen, located in Ann Arbor, Michigan. Today, I will be discussing extractables and leachables studies of electronic nicotine delivery systems. The FDA requires extractables and leachables studies for pre-market tobacco applications. However, little guidance or precedent exists on how to approach these studies. In particular, extractables and leachables on electronic nicotine delivery systems, more commonly referred to as vaping devices or vapes, present some unique challenges for E&L. Vaping devices used in these studies were all pre-filled disposable units. Components of vaping devices were sent individually from the manufacturers. To find out where the individual pieces of the vapes were located in the flow path, a representative device was dismantled. Individual pieces were numbered. From this example image, we can see that there is very little separation of the e-liquid from the other components in the device. The e-liquid is therefore in contact with almost every piece of the device, meaning none of the pieces are low risk in terms of extractables and leachables. Materials of construction were determined using FTIR. As there was overlap in some of the materials, these could be considered a single component. Interestingly, in this example and in several other vaping devices, the part that was labeled as cotton for e-liquid storage was actually polyethylene. There was real cotton inside the device, but this was restricted to part 15, a small piece of cotton attached to a polysilicone cylinder and PVDF-coated wires. Before embarking on an extractable screening, analytical evaluation thresholds, AETs, were calculated for the components. The safety concern threshold was set at 1.5 micrograms per day, and the maximum daily dose for e-liquid was set at 10 mL per day, based on toxicologist's recommendation. As this particular device had a maximum fill volume of 3 mL, the AET was calculated to be 0.45 micrograms per component. The AET was further converted to micrograms per mil for the extraction preparation. In this example, 20 components were extracted in 45 mL of solvent, resulting in an AET of 0.2 micrograms per mil. After determining the materials of construction and calculating the AET, extractable studies were performed on the components under exaggerated conditions. A set of components was weighed, placed in a round-bottom flask, and refluxed for 8 hours in purified water. Using different sets of components, the process was repeated for isopropyl alcohol, hexanes, and 2% nitric acid. Controls were prepared for each solvent by refluxing for 8 hours without components. In this particular study, organic extractables were analyzed in polyethylene e-liquid storage cotton, the polycarbonate mouthpiece, the silicone plugs, and the real cotton. Elemental extractables were further assessed in the PVDF-coated wires and the aluminum vape body. Extracts underwent the following analyses. First, gravimetric analysis was performed to determine the non-volatile residues for each extract, as well as to concentrate the samples for HPLC analysis. The 2% nitric acid extracts underwent elemental analysis of ICP-MS. Semi-volatile and volatile extractables were quantified with GC-FID and characterized with GC-MS. Non-volatile extractables were quantified with HPLC-DAD and characterized with LCMS. This representative GC-FID chromatogram compares the hexane control and the hexane extract of polyethylene. Extractable peaks above the AET of 0.2 micrograms per mil are integrated. Using relative retention times of a butylated hydroxytoluene standard as a marker, the same extractable peaks were found in the GCMS analysis for characterization. This example shows the GCMS total ion chromatogram of the hexane extract of polyethylene. Extractables were tentatively identified to the extent possible with the assistance of the NIST library search. This example shows a tentatively identified slip agent in the hexane extract of polyethylene. Non-volatile extractables were quantified with HPLC-DAD. Extracts and controls were concentrated tenfold for HPLC analysis. This example shows the isopropyl alcohol control and the isopropyl alcohol extract of silicone at a wavelength of 250 nanometers. The extractable peaks above the limit of detection and AET are integrated. Extracts and controls were further analyzed by LCMS for characterization of the non-volatile extractables. This example shows the total ion chromatogram in the top panel, with the background subtraction region highlighted in the green box. The middle panel shows the mass spectrum of the extractable peak around 36 minutes in the isopropyl alcohol extract of silicone. The lower panel shows the diode array detector total wavelength chromatogram, which assisted with finding the correct peak for characterization. Prominent ions were then compared to an in-house polymer additive library for tentative identification, but in this case there was no reliable match. This table summarizes the total number of organic extractables above the AET of 0.2 micrograms per mil for each component. Water extracts contain the fewest extractables, most likely due to the polarity of the solvent. The only extractables above the AET in water extracts were non-volatile and specific to polyethylene. Hexane extracts were analyzed for semi-volatile extractables only. For this solvent, silicone contained the most peaks above the AET, whereas polycarbonate contained the fewest. Isopropyl alcohol extracts were analyzed by gas and liquid chromatography. In the gas chromatography analysis, the silicone also had the most peaks in the isopropyl alcohol extraction, whereas the isopropyl alcohol extract of polycarbonate contained the most peaks in the liquid chromatography analysis. Notably, the real cotton contained no organic extractables above the AET. Extractables were tentatively identified to the extent possible. Most extractables were oligomers of the polymeric components. Slip agents such as stearic acid were found in most of the extracts. However, identification was challenging due to the high background noise in the spectra. All extracts contain metal extractables above the limit of detection, with the aluminum vape body unsurprisingly containing the highest number of metal extractables. Interestingly, the real cotton had more metals than the synthetic components. This table lists metals above the AET for each component. For the metal components, ICP-MS results confirmed the vape body was primarily composed of aluminum, and that the wires were mostly made of copper. Of those metals above the AET, most had relatively low toxicity. Generating the full library of potential leachables under stress conditions, the extractable studies informed the leachable studies of the e-liquid. A general approach was taken to Leachables method development to ensure sensitive and selective methods for Leachables analysis. First, the AET for Leachables was calculated to determine how to prepare the e-liquid samples. Notably, the e-liquid used in method development was a virgin sample that had not been in contact with the vape device. In cases where there are multiple flavors, the flavor with the lowest background was selected for method validation. However, data from other flavors was useful for sample testing to rule out excipients from the e-liquid flavoring. Generally, tobacco flavors had the lowest background. To simulate the worst-case leachables, extracts from the Extractables study were spiked into the e-liquid samples to determine if the extractable peaks are well resolved. In general, two methods were developed and validated for monitoring of leachables during routine stability testing. Both are mass spec compatible, in case a leachant needs to be identified in the future. A GC-FID method monitored semi-volatile and volatile leachables. Butylated hydroxytoluene, Ph.D, was used as a representative semi-quantitative standard. Non-volatile leachables were monitored with HPLC-DAD at wavelengths of 210, 250, and 280 nanometers. Here, diphenylamine was used as a representative standard. Based on the safety concern threshold of 1.5 micrograms per day and the maximum daily dose of 10 mL, the AET for leachables in e-liquid was 0.15 micrograms per ml. Example preparations of e-liquid for GC and HPLC are outlined. E-liquid was prepared for GC by extraction of dichloromethane. E-liquid was prepared for LC by extraction in dichloromethane, drying under nitrogen, and reconstituting in isopropyl alcohol. These example chromatograms demonstrate specificity of the gas chromatography method for potential leachables from polyethylene. The top panel shows the hexane extract of polyethylene from the Extractables study, zoomed to show the extractable peaks. The bottom panel shows the e-liquid extract. The middle panel shows the hexane extract of polyethylene spiked into the e-liquid sample as a surrogate worst-case leachables. The extractable peaks are still well resolved in the spiked sample. Similarly, this example shows the specificity of the HPLC-DAD method for analysis of non-volatile leachables from silicone in e-liquid. The top panel shows the isopropyl alcohol extract of silicone at a wavelength of 210 nanometers, and the bottom panel shows the e-liquid sample. The middle panel shows the silicone extract spiked into the e-liquid and illustrates that the extractable peak from silicone is well resolved. Some flavors pose more difficulty for leachables method development than others. For example, this is a GC-FID chromatogram of a banana mint flavored e-liquid from another study after cleanup with liquid-liquid extraction. There are many excipient peaks, which can interfere with the analysis of leachables. For that particular study, peach mint flavor had the least interference. Even then, there are excipient peaks in the sample, but less overlap with extractable and leachable peaks. A similar issue was also seen with certain flavors in HPLC-DAD. This is a chromatogram of pomegranate berry mint flavored e-liquid at UV wavelengths of 250, 210, and 280 nanometers. Again, many excipient peaks were present, which could interfere with analysis. In this study, the peach mint flavor was found to have the least interference for HPLC-DAD analysis of non-volatile leachables. However, it is still apparent that there are many peaks in the e-liquid. This chromatogram shows the e-liquid sample, concentrated 40-fold for analysis, at wavelengths of 250, 210, and 280 nanometers. Leachables methods were then validated following ICH Q2 guidelines to prove suitability for the monitoring of leachables in e-liquid at the AET of 0.15 micrograms per mil. The following parameters were assessed: linearity, accuracy, method precision, intermediate precision, limit of detection, LOD, limit of quantitation, LOQ, robustness, and specificity. One of the parameters of the method validation was linearity. This example shows a linearity curve for Ph.D. from the validation of a GC-FID method for volatile and semi-volatile leachables. The linear range of the method was 0.2 micrograms per mil to 50 micrograms per mil, with a correlation coefficient between peak area and concentration over 0.99. This table shows the results of accuracy and precision testing for the validation of a GC-FID method for the monitoring of volatile and semi-volatile leachables in e-liquid. Ph.D., a representative leachable standard, was spiked into e-liquid at 3 different concentrations covering the linear range of the method. The method limit of quantitation was 0.2 micrograms per mil, and at this level, the average percent recovered from e-liquid was 126. This met the acceptance criteria of recovery between 70 and 130%. 6 replicates were prepared for the accuracy and precision testing at 10 micrograms per mil spike level. Meeting the acceptance criteria for accuracy, the average percent recovery was 116. The percent relative standard deviation of the 6 replicates was 5, which was well below the acceptance criteria for precision of 15. Spike recovery was also assessed at 50 micrograms per mil, which was the upper linear range of the method. The average percent recovery was 89 at this level, which also met acceptance criteria for accuracy. Similarly, the linearity of the HPLC-DAD method was assessed by a diphenylamine standard at concentrations ranging from 2 to 50 micrograms per mil. This graph shows the linearity curve at 210 nanometers, with a good fit between the concentration and DPA peak area. As the correlation coefficient was not less than 0.99, acceptance criteria for linearity were met at this wavelength. At 250 nanometers, the method was also linear from 2 to 50 micrograms per mil. The best fit between concentration and peak area was at 280 nanometers. Here again, the correlation coefficient was not less than 0.99. Thus, acceptance criteria for linearity were met at all 3 wavelengths, despite 280 nanometers being slightly better than the other 2. Similarly, accuracy and precision testing was performed for the HPLC-DAD validation by spiking diphenylamine into e-liquid at levels covering the linear range of the method. This table shows data from the 210 nanometer wavelength. The method limit of quantitation was 2 micrograms per mil, and recovery at this level was assessed in triplicate. Method accuracy and precision was tested at the 10 micrograms per mil spike level using 6 replicates. At the upper range of the method, 50 micrograms per mil, recovery was assessed in triplicate. Meeting the acceptance criteria, the mean percent recovery was between 70 and 130 at each spike level. For precision, the percent relative standard deviation of the 10 microgram per mil spike was 2, well below the acceptance criteria of 15. After validation, methods were used to test samples for leachables. This example GC-FID chromatogram shows a peppermint-flavored control, which was not in contact with the packaging, and a stability sample, which was also peppermint-flavored. Here, there were no leachables found in the sample. Difficulty arose when the control was not the same flavor as the sample. However, for this particular client, a follow-up study used GCMS and LCMS to determine which peaks were related to excipients and which peaks were true leachables. It turned out there were no leachables above the AET. More data should be available soon, but most of the Leachables methods are still in the validation stage. In conclusion, extractables and leachables for vaping devices presents unique challenges due to the components, device design, and difficult matrix. In particular, non-tobacco flavors contained more interfering peaks in the chromatogram, demonstrating the increased risk of flavored vaping products. I would like to thank Amanda Neely and Brian Gabbett for their hard work on these projects. Next, I'd like to thank Dr. Derek Beauchamp and Dr. Andrew Colbert for their expertise and advice. Furthermore, thanks to Caitlin Bondar and Katie Morgan with assistance with recording this presentation. Last but not least, I'd also like to thank Smithers E&L USA for hosting this event and giving me the opportunity to present. Thank you for tuning in to this presentation. Are there any questions?